In this video, we're going to discuss the regulation of both glycolysis and gluconeogenesis. So if we recall, when we were talking about glycolysis and gluconeogenesis, we said that there were 3 points or steps in glycolysis that had to be bypassed in order for gluconeogenesis to occur. And that was the step catalyzed by hexokinase, the step catalyzed by phosphofructokinase, and the step catalyzed by pyruvate kinase. These 3 steps had very, very large negative delta G values that simply could not be reversed. So we could not simply convert glucose 6-phosphate to glucose using hexokinase in reverse because, again, the delta G is far too negative for that enzyme to be able to operate in the reverse. So we had to bypass this using a separate enzyme in gluconeogenesis. We had to bypass this step using a separate enzyme in gluconeogenesis, and we had to bypass this step using 2 enzymes. To get from pyruvate to phospholipid, we had to use 2 steps to overcome or reverse the enzyme, the step catalyzed by pyruvate kinase. Now, the reason I'm mentioning this is because it's these points in both glycolysis and gluconeogenesis, right here, and right here, that are the 3 major regulatory points for glycolysis and gluconeogenesis. So we want to discuss how the enzymes that catalyze these reactions in both glycolysis and gluconeogenesis are regulated. Because if we want to shut off glycolysis, we're going to do it at one of these 3 points, or all of them. And if we want to shut off or activate gluconeogenesis, it's going to be, again, at one of these 3 points. So here is a figure that shows this in a bit more detail. Again, you may want to pause the video and look at this a little more closely, but essentially we've got glycolysis if we're going down from glucose to pyruvate, and we've got gluconeogenesis if we're going up the slide from pyruvate to glucose. If we look at a couple of these, for example, let's look right here. The enzyme that catalyzes glucose to glucose 6-phosphate is, of course, hexokinase, and glucose 6-phosphate to glucose, the reverse, is glucose 6-phosphatase. These 2 enzymes are reciprocally regulated. In other words, when one of these, say hexokinase, when we want to turn this one off, we want to turn glucose 6-phosphatase on. Or if we want to turn glucose 6-phosphatase off, we want to turn hexokinase on. So they're reciprocally regulated in order to prevent what is called a futile cycle, because we don't want glucose to be converted to glucose 6-phosphate by hexokinase and then at the same time be converting glucose 6-phosphate back to glucose. Again, that would be a futile reaction. We're producing one thing and breaking it down at the same time. And that's just a waste of energy. So both glycolysis and gluconeogenesis must be reciprocally regulated. So when we have one of these enzymes being inhibited, we want the other to be activated and vice versa. That way we avoid a futile cycle and avoid just a waste of energy. So you can see in this figure, we've got a lot of the activators. So for example, what activates phosphofructokinase? We've got fructose 2,6-bisphosphate, ADP, AMP. What inhibits it? The slash represents an inhibitor. The green arrow represents an activator. ATP inhibits this enzyme. Citrate inhibits it. Over here, we saw that fructose 2,6-bisphosphate is an activator. Well, for this enzyme, fructose 2,6-bisphosphate is an inhibitor. So we want to take a look at all of these activators and inhibitors in a little more detail for each of these regulatory points. So this will be our regulatory point here. We have a regulatory point here and here. So let's focus on these regulatory points. We've got, as we mentioned, the major one where both glycolysis is activated and gluconeogenesis is inhibited. The major one occurs at the step catalyzed by phosphofructokinase 1 in glycolysis, and fructose 2,6-bisphosphatase, or excuse me, fructose 1,6-bisphosphatase in gluconeogenesis. So this is our major regulatory point for the two, because as we said, phosphofructokinase 1 is the committed step in glycolysis. So, but then we also want to talk about the regulatory point here with pyruvate kinase and pyruvate carboxylase and phosphoenol pyruvate carboxykinase, that step, that cycle. And then finally, we want to talk about the regulatory point where hexokinase and glucose 6-phosphatase are reciprocally regulated. So let's start off with this middle one here, which is the major regulatory point. So we want to start by talking about phosphofructokinase 1, how it's regulated, and then also then talk about how fructose 1,6-bisphosphatase is reciprocally regulated. So let's start with PFK1, or phosphofructokinase 1. It is a homotetramer, which means it is 4 individual subunits, which all have the same amino acid sequence. This is an enzyme that can adopt a R relaxed or T tense state. So when it's in the R state, it's a much more active state, and in the T state, it's a lower affinity, lower activity state. This enzyme is an allosterically activated or regulated enzyme, which means, of course, that we have small molecules which bind at regulatory domains and either activate it or inhibit it. So a couple of inhibitors, represented by the negative sign, citrate and ATP, when they bind to the enzyme, they inhibit it or lower its activity. They would probably stabilize the T state, giving it a lower activity. And these molecules, citrate and ATP, generally those are abundant when the cell has a lot of energy. We'll talk about citrate as an intermediate in the citric acid cycle, and of course ATP is our energy currency. So when these are high, we do not want to continue on with glycolysis because we've already got enough energy. So these tend to inhibit this enzyme, therefore inhibiting glycolysis. AMP and ADP, those are activators. These are produced when ATP is hydrolyzed. So when these are high, it means we don't have a lot of ATP. So these bind allosterically and shift our equilibrium to the R state, increasing our activity and our affinity for our enzyme for the substrate. And then one we want to talk more about is a molecule called fructose 2,6-bisphosphate, which is a potent activator of PFK1 and therefore an activator of glycolysis. So we'll talk more about this in just a little bit. Let's look at a couple of activity curves here. Here we have PFK1 activity versus its substrate concentration, fructose 6-phosphate. When there's no fructose 2,6-bisphosphate, remember this is the activator of our enzyme. When this is not present, we have a sigmoidal shaped curve, but when it is present, when we begin to add it to our solution, it binds allosterically, shifting our curve to the left. Okay? Now that causes our Km to decrease, which means we have a higher affinity for our substrate, and therefore when it's not present, we have a higher or greater Km value, giving us a lower affinity for our substrate. Again, our substrate here is fructose 6-phosphate. Let's look at the curve for ATP binding. When there is low ATP concentrations, our enzyme tends to be more in the R state. You can see we have a lower apparent Km here, giving us a higher affinity, and we have a much greater activity. When ATP is present, it binds allosterically, and it tends to shift our curve to the right, again making it more sigmoidal, stabilizing the T state, and giving us a higher Km, indicating a lower affinity for our substrate. So those are some activators and inhibitors of PFK1. So, well, what about fructose 1,6-bisphosphatase? This enzyme participates in gluconeogenesis, so this is a major regulatory point in gluconeogenesis. Common regulators, AMP inhibits it, and of course we said that was an activator of PFK1, and fructose 2,6-bisphosphate is also an inhibitor. So let's talk about fructose 2,6-bisphosphate because that's really a key regulatory molecule for both enzymes and therefore a key regulatory molecule for glycolysis and gluconeogenesis. So here is a figure you may have seen before, which shows us the steps in glycolysis. I want you to look right in here. Here we have fructose 6-phosphate being converted to fructose 1,6-bisphosphate, of course, by the enzyme phosphofructokinase. Now, there's a side pathway or a kind of an offshoot of this pathway which can occur. Fructose 6-phosphate, which we see here, can be converted to fructose 2,6-bisphosphate. Not fructose 1,6-bisphosphate, but fructose 2,6-bisphosphate under certain conditions. Now remember, this is that key regulatory molecule which activates PFK1, inhibits fructose 1,6-bisphosphatase. So let's talk about this particular molecule, how it's produced, and how it regulates the activity. So let's say that our glycolysis is going along and we turn glycolysis off. Okay, so remember right here is a key stopping point or regulatory point for glycolysis. So let's say we inhibit PFK1. What's going to happen to the levels of fructose 6-phosphate? Of course, over time they're going to start to build up, right? Because we've turned off glycolysis. So fructose 6-phosphate levels begin to build up when glycolysis is turned off. Well, when fructose 6-phosphate begins to build up, it's going to shift our equilibrium over here to the left, which means we're going to start producing more fructose 2,6-bisphosphate. Okay? What happens when fructose 2,6-bisphosphate is produced? Well, as we start to increase levels of fructose 2,6-bisphosphate, those levels, as they increase, they begin to bind to PFK1 and activate it. Right? We said it's a potent activator, so this molecule binds to PFK1 and activates it. And also, when this molecule builds up, it binds and inhibits fructose 1,6-bisphosphatase. Right? So as this begins to build up, what happens? PFK1 begins to get more active. Fructose 1,6-bisphosphatase begins to be inhibited. And what's the net effect? Well, glycolysis is going to start to become activated because PFK1 is active and gluconeogenesis is going to be inhibited. So when we turn off glycolysis, over time, the levels of this molecule will begin to build up, and that's going to cause glycolysis to begin to turn back on and gluconeogenesis to be turned off. Okay. Well, what about when fructose 6-phosphate levels are low? Okay, so let's say that we've got glycolysis is running very quickly and we begin to deplete our levels of fructose 6-phosphate. Okay, so we have low concentrations of fructose 6-phosphate. Well, that's going to shift our equilibrium from left to right. So this molecule is going to start to be broken down to form fructose 6-phosphate. So that's going to cause the levels of fructose 2,6-bisphosphate to drop, right? Well, what effect will that have? Well, when fructose 2,6-bisphosphate levels begin to drop, then we're removing that activator of PFK1. So PFK1 will not be active. It'll have a low activity. Fructose 2,6-bisphosphate, fructose 2,6-bisphosphate levels, when they again begin to become low, fructose 1,6-bisphosphatase levels, that enzyme is no longer going to be inhibited because this is an inhibitor of that phosphatase. So we're removing the inhibitor, so that's going to cause the enzyme fructose 1,6-bisphosphatase to begin to become more active. Well, what's the net effect? Gluconeogenesis is going to begin to become more active, and glycolysis is going to begin to become less active. So the levels of this molecule right here really determine whether we're going to have glycolysis is going to be our major pathway or gluconeogenesis is the major pathway. So be sure to take some time and go over this a few times. I know it'll take a few times to really make it clear, but understand how the levels of this molecule are control glycolysis and gluconeogenesis by controlling those two key enzymes. Now, since this is such a key molecule, what is the enzyme that produces it or breaks it down? Okay. So we need to talk about the enzyme that is responsible for producing fructose 2,6-bisphosphate and also the enzyme that is responsible for breaking it down. And it turns out it's the same enzyme. It's a very unique enzyme. The synthesis and degradation of this key molecule are controlled by or catalyzed by a single enzyme. This enzyme has both a kinase, so it can phosphorylate our fructose 6-phosphate, and a phosphatase activity, so it can remove that phosphate and regenerate the fructose 6-phosphate. The enzyme that does this is what we call a bifunctional enzyme because it has both kinase and a phosphatase activity. And so it's often called PFK2, phosphofructokinase 2, but it's more correctly or completely called phosphofructokinase 2 fructose 1,2,6-bisphosphatase 2. Okay, so this bifunctional enzyme has a kinase and a phosphatase domain, so it can phosphorylate our substrate to produce that key regulatory molecule, or it can break it down by removing a phosphate and therefore control glycolysis and gluconeogenesis. So this enzyme, which we'll just call PFK2 here for short, we have to also appreciate how it is regulated. Since this enzyme rather, produces the molecule that activates or inhibits glycolysis, gluconeogenesis, how is this enzyme PFK2 regulated? Here you can see one other slide showing that this enzyme is a single polypeptide chain with a kinase and phosphatase domain. So let's talk about the regulation of this PFK2. It is controlled ultimately by a molecule called glucagon. Glucagon, let me jump forward here just a bit. Glucagon is a peptide hormone, contains about 29 amino acids secreted by the pancreas. Glucagon binds to a receptor predominantly on the liver and controls the activity of our PFK2. So let's take a look at what happens there. So glucagon, secreted by the pancreas, it binds to a receptor, and that receptor activates an enzyme called adenylyl or adenylate cyclase. Adenylate cyclase takes ATP and produces the second messenger, cyclic AMP. Cyclic AMP, as it begins to get produced, it's a very potent second messenger, and it binds to a kinase called protein kinase A, or pKa. Here you can see cyclic AMP binding pKa and activating. So glucagon activates adenylate cyclase. Adenylate cyclase produces the second messenger, cyclic AMP. Cyclic AMP binds and activates pKa, and pKa is a kinase, so it phosphorylates our bifunctional enzyme. When it phosphorylates it on the kinase domain. And when it phosphorylates on the kinase domain, which you can see here, the phosphatase is inactive, or excuse me, the phosphatase is active. So by phosphorylating the kinase domain, it inhibits the kinase domain and activates the phosphatase. When it's not phosphorylated, so when there's no glucagon, pKa is not active, and this is not phosphorylated, then the kinase domain is active. So what happens when the kinase domain is phosphorylated? Well, we get the phosphatase, which means we're going to break down our fructose 2,6-bisphosphate. And when glucagon is not present and this is not phosphorylated, PFK2, the kinase, is active, which means we're going to phosphorylate fructose 6-phosphate and produce our fructose 2,6-bisphosphate. So let me go through those steps again because I know that's a lot to take in. Let's start with our glucagon. So glucagon is released from the pancreas, and it's released in response to low blood glucose levels. So when we need glucose, we need to get glucose into our blood, maybe we've been fasting or exercising a lot, we need glucose to be delivered to our bloodstream. The pancreas will release glucagon. Glucagon binds to a receptor on the liver cell. The liver cell, when the membrane is active, then it activates adenylyl or adenylate cyclase, which produces cyclic AMP. Cyclic AMP then does what? It binds to pKa and activates pKa. And of course, pKa is a kinase, so it phosphorylates our bifunctional enzyme. When it phosphorylates the enzyme, the phosphatase is active. And when the phosphatase is active, our fructose 2,6-bisphosphate levels will drop because we're removing one of our phosphates. We're going to take off a phosphate and convert that to fructose 6-phosphate. Well, what happens when this molecule drops? Remember, it's going to do what? It's going to decrease our phosphofructokinase activity because that's an activator. So our PFK1 activity will drop and our fructose 1,6-bisphosphatase levels will increase. So essentially we're going to decrease flux through glycolysis and stimulate gluconeogenesis. Well, gluconeogenesis is going to produce glucose, which then can be secreted into the bloodstream, giving us more glucose for our brain to use. So again, there are several steps, but we're going to be sure we appreciate from glucagon all the way how that affects our bifunctional enzyme, how that affects our key regulatory molecule, our fructose 2,6-bisphosphate, and how that ultimately affects PFK1 and fructose 1,6-bisphosphatase, which ultimately affect glycolysis and gluconeogenesis. So be sure to take some time, maybe draw out a flow chart, and be sure you understand the regulatory steps that occur at this key regulatory point in glycolysis and gluconeogenesis.